Sagi Snir from Italia.
Okay, so as you see, the title is Maximum Parsimony on Phylogenetic Networks and Some Rigorous Results. And horizontal gene transfer is the transfer of genetic material from one organism to another, not through linear descent. And it's, wow. Yeah. Well, I see the effects have vanished. But OK, so it's very common among microbes, and it's a significant factor in how bacteria develop resistance to antibiotics. And the graphical representation is the introduction of this edge along the phylogenetic tree. Now, Tandy Warnow spoke about the importance of lateral gene transfer in the tree of life. And if we use the very impressive figure of Bill Martin, so if we look at the history of the evolutionary history of the DNA in the universe, so even if we focus on a very tree-like portion, which is here, if we use a magnifying glass, we see a constant movement of DNA segments from one linear to another, which is not displayed here. So to show the magnitude of this phenomenon. Now, when we come to analyze lateral gene transfer, so a key observation is as follows. Although the history, the evolutionary history is displayed or described by a network, every single gene is actually evolves along a single tree. And if we use this illustration that illustrate phylogenetic networks over 4 taxa, and each taxa is composed of 2 genes, the blue gene and the red gene. So in practice or actually, every gene evolves along a single tree, where the red gene evolves on the organismal tree. But the blue gene, this organism, inherited the blue gene by a lateral gene transfer from here, and actually its ancestor is this guy. Okay, so we actually get this tree. Now, in this talk, I will focus on combinatorial aspects, which is the parsimony criterion. And let's review very briefly the parsimony criterion. So given a topology, a tree, and sequences at each node in the tree, the parsimony criterion is actually the sum of the Hamming distances induced by the sequences in the nodes. And if we look at this binary example, so we get Hamming distance 1 along this edge, 0 here, and 2 here. And we get score 3. Another example, totally different topology and labeling. And the score here is 4, is 2, 0 here, 0 here, 0 because you see 0, 0 here, and 0, 0 here. 1 here, and score 2. Now, the maximum parsimony problem, the small MP problem on trees is as follows. Here, the input is a little bit different. We don't have the sequences on internal nodes, rather only a topology and strings on the leaves. And the task is to find the optimal assignment on internal nodes that leads or yields the minimum parsimony score as defined before. Okay, so this is one labeling that induces this Hamming distance, and the score is 3. Another one, which is 1, 1, okay, 1, 1 here, 1, 1 here, and we get score 4. And this one, 0, 0, and 0, 0 at internal nodes, and we get score 2, which is optimal. Now, this problem is solved by a classical bottom-up dynamic programming algorithm by Walter Fitch that actually devised this criterion in 71. And now let's see how we extend the MP problem to networks. So it actually is based on Utenheim extension for MP on recombination networks, and the extension is as follows. The organismal tree is assumed to be known, and we want to accommodate the given gene by adding the least number of lateral gene transfer edges. So the last definition I will give is what are the network trees. So the network, a tree is obtained or induced from the network by a series of edge deletions. And like we see here, so in this tree, we deleted this edge, while in this tree, we deleted the edge that went here. So we got 2 trees. So as probably we all speak about the same things, and we all use different terminology, so I think Stephen Wilson said it as well, that this number grows exponentially with the number of lateral gene transfer edges, and so it's quite straightforward. And now we can define small MP, small maximum parsimony on networks. And so the input is a network and a set of species. Okay, so the network and on a set of species with the sequences, the genes, okay? And the task is to find for every single site which is the tree that minimizes the parsimony score. To find the small parsimony, maximum parsimony problem for every tree. Okay, so let's see a simple example. So here we got the network, so these are the trees, and we now try to solve small MP on every character. So we look at this one, and we can take the red tree, and we put 0, 0 here and 1 here, and a single change on this edge. Again, here, this is the same character, so we use the same tree and same labeling. This is a different character, but again, with the same tree and different labeling, internal labeling, 1, 1, 1, we get single change on this edge. So still, we are optimal. Now, at this character, 1, 0, 0, 1, there is no way in the world we can achieve a single change with the red, but we can use this tree, the blue tree, and put 0, 1, 1 here, single change on this edge. Same character, different character, but still using the blue tree, we get a single change on this edge. Okay, so small MP on networks, what is the result? This is unexpectedly is MP hard, in contrast to small MP, which is solved by Fitch algorithm. By the type of reduction, we can also infer that this is hard to approximate to any constant factor, but it allows a recursive dynamic programming exact algorithm that I will talk next. And it improves an exhaustive algorithm by Nachle et al. That actually exhaustive, that it tried every tree in the network. So what is the so what is the type overview of our dynamic programming algorithm is as follows. So again, different, we all say the same things. Daniel Husson, I don't remember what was his terminology, but we call it a conflict. So a network is given, and we seek for a tree. Okay, we seek for a tree in the network. It needs to be an optimal tree. Okay, a node with integrity bigger than 1 is a conflict because we look for a tree. Okay, so this is a conflict. A conflict is removed by selecting exactly one incoming edge and removing all the rest of the edges, whether it is LGT or tree edge. Okay? We wish to remove all the conflicts recursively until we end up with an optimal tree. Okay, so how it goes? So apply recursively the following procedure. So again, like Steven said, there is some, we can define some invariants on this network that there is a lowest edge, okay, by the partial relation on the tree. So there is a lowest edge, which is an edge that there are no lateral gene transfer edges below it, underneath it. So we locate one, and we solve the conflict that is induced by that edge, and we apply it recursively until we end with a tree. So here is a lowest edge, and this is the conflict that induced by it. So we solve this conflict by remove this edge. Another lowest edge, and this is the conflict. Another one, we remove this. And now last conflict is here. We remove this edge, and we remained with a tree. Whether this is optimal, I can't promise you. OK, now what is the procedure? And remember, this is, supposedly there is no efficient algorithm. So what is the procedure for every lowest edge? So there are two simple cases where the decision is easy, and I cannot say the details in the talk, but I'd be happy to say it later. So it's easy. So either we remove the tree edge like this one and retain the lateral gene transfer edge, okay, according to parsimony criteria, or we remove this edge and remain, and a conflict is solved. But there is the problematic case where the decision is hard, and here I need to check both directions, either to remove this edge and continue, and recursively remove this edge and try this track, okay? And this accounts for the exponential factor. However, we can say few rigorous things, which is this. The number of problematic cases is linearly bounded by the optimal score, the optimal parsimony score of the optimal tree. Okay, and this leads to this corollary that in almost all practical cases, the algorithm you just saw is fixed parameter tractable when the parameter is the optimal score. Okay. And another corollary is that simple twist to this algorithm that I showed yields a three approximation algorithm that is based on local ratio variant, which I'd be happy to speak about, but I want to detail some results on data that we have. So remember, we just spoke about the small problem, but actually we try to solve the big problem, and the big problem is as follows. We need to add edges to the tree until we get to the optimal score. Now, key observation, it never harms to add more and more edges. Your parsimony score never get worse because you just add more and more trees, and you just increase your selection. So, this leads to parameterized definition that find the best network with k edges. And in practice, we just use a heuristic that we always expand the best network. And we don't have, for this, we don't have some rigorous results, whether this is the right approach. But it led to good results. Now, I want to discuss some biological findings that we had. So the Rubisco gene is in charge of carbon fixation in cells, and it's a key enzyme in photosynthesis. And actually, it's the most abundant enzymes on Earth, and which I heard from guys in the lab that it is very crucial today for production of wheat and stuff like that. Now it divides to red and green like Rubisco based on the plants or on the organism it is found with. So. So the Rubisco tree for the gene tree of the Rubisco for these guys, these organisms, is incongruent with the species tree of these organisms. And Delwiche and Palmer in MBE in '96 claimed that at least 7 lateral gene transfer events are necessary to explain how the Rubisco tree evolved, okay? So they hypothesized 4 lateral gene transfer events between the green and red, but with the type, the form 2 Rubisco, they could not cope. And this is their Rubisco tree, the gene tree of the Rubisco. Here we see the green type Rubisco. We see the red type Rubisco, and here is the form 2 Rubisco. And these are the organisms. Here we see the rice and the nicotiana, which is the nicotine. And these are the red algae, okay? And actually, this is a convexly colored tree where you see that the green, all the green are one subtree, the red is subtree, and the form 2 is also a subtree. And here is the species tree of these organisms, okay? And you see that this one is completely not convex with respect to this coloring, the red and the green. And what is their explanation? So they did not cope with the Rubisco 2, but what they did is to collapse every monochromatic subtree into a single leaf, and now they pass lateral gene transfer edges also with some other knowledge to explain the incongruity, and they did it manually. Our explanation is as follows: we run the big problem, okay? We added edges in the procedure I spoke to you, and these are the edges. So here we explain all the tree, and we got exactly the 7 events that they hypothesized. The resolution is high, is between single branches and not between groups, okay? And it's automatic. It's the procedure that we developed. And this is how the parsimony score goes, bunches. We see that after 7 events, the improvement decreases. Here we did not apply the criteria that Barbara spoke about, like BEAK and the AIC that we now, or some other combinatorial, but we now developed them. And let me just give acknowledgement. So all the results are from these papers, and all these papers were with Guajin and Luai Nachle from Rice University and Tamir Tuller from Tel Aviv University. And let me, like Olivier said, that we need to move from combinatorial to statistics. So these are all the works, and now our efforts are really in the statistical realm to apply maximum likelihood and hidden Markov models to phylogenetic networks.
Maybe we'll carry on the discussion over coffee, and we'll be back here at 11:30. Please make sure you've got your talks on the machine beforehand if you can.
Okay, so sorry.